Welcome to the INDEPTH Academy webinar. My name is Celia Baroux. I work at the University of Zurich at the Institute of Plant and Molecular Biology. I would like to tell you today about our work on linker histones, and I will embed it in a bit of a broader context. What do we know about linker histones in plants? And we mostly work with Arabidopsis as a plant model. Okay, so histones are a major component of the nucleus. They were discovered already in 1884 by Albert Kossel. At that time, the existence of nucleic acid has just been discovered, and it was still people were still in the search of how heredity could be conveyed from one generation to the other, how the genetic information was organized in the cell. And a lot of biochemical experiments were conducted at that time to extract the basic components of the cells and of the cell nucleus. And Albert Kossel discovered that you could extract a very massive fraction of proteins under really harsh acid and salt salty buffer. So there's an original paper in German that actually explains that he decided to turn these protein histones based on the fact that they have peptone-like biochemical properties in this extract. And later on, it was discovered that this histone substance is not constituted by a single component, but by several components. So running the extracts of histone on a protein gel allowed to separate different components of this histone fraction. And the histone components were named after their appearance on the gel. So with a band running here on top in a 2 isoform called H1, and then there was H2, H3, and H4 components that were on the gel. For a very long time, actually, epigenetics has been focusing on the histone H2A, H2B, H3, and H4, which constitute the core nucleosome. And histone H1 has been very long, I would say, ignored or not really considered. So it has been long forgotten. But in the past decades, there has been a resurging interest into this histone, and especially given its emerging role in regulating gene expression. So linker histones are part of the chromatin together with the core nucleosome histone. I'm not going to teach you anything saying that the chromatin is built as a polymer where DNA is wrapped around core nucleosome, and each core nucleosome is constituted by an octamer of histone, H2A, H2B, H3, and H4. And this bits on a string structure is further coiled and folded in a more complex fiber. And the level of folding of this fiber has long been thought to be very regular throughout the nucleus. It was thought to have especially a supercoiled or super folded level of 30 nanometer. And this super folded level has been challenged quite recently. So we now understand that it's a bit more complicated than that. And in this organization, in this folding organization, linker histone have been attributed a role mostly of rather stiffener and architect. So the nucleosomes themselves were very long thought to be rather inert and structural chromatin components, and due to their very massive organizations, they were thought to be a hindrance to transcription. So for a long time, they were simply thought to be repressors of transcription. But nowadays, we know that they have multifaceted roles, and they are actually fine-tuning gene expression, especially through epigenetic modification. And the linker histones themselves were long thought to be also purely structural components that would actually stiffen the chromatin fiber. What you see on the top right of this slide is an electron microscopy micrograph of a chromatin fiber that has been extracted from the nucleus. And you can see on the left side the bits on a string structure that is typical of a diploid chromatin fiber without linker histone. And on the right side, you can appreciate that this chromatin fiber, which now has been incubated with linker histone, has a very specific appearance, especially a tic-tac appearance. And what is happening here is that the linker histones are imposing an angle at the exit of each nucleosome. And the overall architecture, which is gained by the addition of H1, gives the impression of a stiffened chromatin fiber. So more recently, through modern technologies, the same configuration of linker histone with respect to nucleosome has also been visualized with a particular type of microscope, so atomic force microscopes. And where you can see here, these bright spots that you have here constitute a nucleosome with a linker DNA in between. And nucleosomes are rather far apart in an extract which doesn't contain any linker histone. As soon as you add linker histone in the extract, you can appreciate that the nucleosomes are forced coming together. And this is, of course, not an all or none response, but it is a biophysical process which is a little bit variable, but it is definitely constrained with linker histone. So the distance between nucleosome is definitely reduced and constrained as soon as you have linker histone. This is now schematized here. So linker histone are proteins which are binding at the exit of the nucleosome, and by doing so, they bring together linker DNA on each side of the nucleosome and brings them together. So it ends up in a configuration where nucleosomes come very close together and are compacted. So for a long time as well, we were left with this idea that linker histone are responsible for chromatin compaction mostly. So linker histones are architects of the chromatin fiber, but at a higher order folding level. In Arabidopsis, there are 3 genes encoding for linker histones, named H11, H12, H13, and they have been largely analyzed by the group of Andrzej Germanowski at the University of Warsaw. And his group has characterized in detail the property of the different linker histones. So what you can see on the left graph here is that is a so-called FRAP experiment for fluorescence recovery after photobleaching. And this experiment allows to measure the mobility of linker histone on the chromatin. And the steeper is the curve, the more mobile is the linker histone. What you can appreciate is that the 3 variants are slightly different in terms of mobility. H13, the green curve, being the more mobile, then comes H11 and then H12, which has a slightly longer residence time. So in Arabidopsis, we have these 3 variants. We know that they locate everywhere in the nucleus, meaning that they co-localize with both Euchromatin and Heterochromatin compartments. At the molecular level, the distribution of linker histone has been mapped by a CHIP experiment. And we know that in heterochromatin, they are enriched over transposable elements. So let them be heavily heterochromatic transposable elements that are localized near the centromeres or more distally localized transposable elements. In Euchromatin, H1 is also distributed along the genome. So I would like here to take the opportunity to correct for an old-fashioned view, which is that H1 is only associated with heterochromatin. No, H1 is also located in Euchromatin. And in Euchromatin, however, when you do molecular profile, you can see that H1 anticorrelates with an epigenetic mark called A3K4 trimethylation, which is associated with permissive transcription. So in other words, the more the gene is expressed, the less H1 you have in its vicinity, but it's not that it's completely absent. And as a matter of fact, if you correlate the presence of H1 with expressed genes, and if you classify the gene depending on their expression strengths, you can see that H1 is present among all these classes, but it is definitely anticorrelated with the expression level of the genes. So there is still a trend which matches the former view that H1 correlates with gene repression, but it's not a component which is absent or present with expression or no expression. There is a bit more of a modulated correlation here. So I will mostly talk for the rest of my slides about what we know in general terms about the major variant, which are H11 and H12. I will not talk so much about the variant H13, has been described in detail by the group of Arabidopsis. And H13 is a variant which responds specifically to stress, to drought stress and light stress. It is a very interesting variant, but I will not talk about it today. Okay, so H11 and H12 are the so-called canonical variant, and they are very similar at the structural level. They are also expressed in the same tissue. However, when we look at their distribution at subnuclear scale, we see that they still localize to distinct regions. So what you have here is a single nucleus, which has been split in 3D along different orthogonal planes. And here you have regions zooming inside the nucleus. And again, another inlet showing the distribution of the two variant H11 and H12 with two different colors, either around the chromocenter or in Euchromatin. So you can see that in chromocenter, H11 RFP seems to be mostly localized at the periphery, whereas the interior of the chromocenter, so this dense heterochromatin region, H12 is prevalent. And in Euchromatin, we have an intermingling of regions which are enriched in either H11 or H12. So molecular profiles are still on the way to try to distinguish whether H11 or H12 have preferential regions where they would bind, but definitely at the nuclear level, you can see that we have isolated or very distinct regions decorated by these two distinct variants. Also at the tissue scale, what we can see is that H11 and H12, even though they are structurally really similar, they differ by only a few amino acids, and they have similar property, except that H12 is slightly less mobile than H11. What we see is that there is an opposite correlation of their abundance depending on the differentiation level. So you have here a picture of a root tip in Arabidopsis, and below you have a cell file going from the left to the right, from the less differentiated to the more differentiated state. And as you can see, that when we go more towards differentiation, we increase the abundance of H12 at the detriment of H11. So here again, it points out towards different function of H11 and H12, and but we don't know exactly which kind of function. So there's more investigation which are on the way. Now, a few years back, we were asking a much more general questions, because we knew that in many organisms, the depletion of H1 can be lethal, especially in mammals, in yeast, and in worms. It is not necessarily lethal, even though the organism have very strong defect. But we were wondering whether Arabidopsis would actually support the depletion of H1. So the group of Andrzej Czemanowski had done already an RNA interference line, which was downregulating massively the 3 linker histone in Arabidopsis, and they found really severe phenotype with a lot of abnormal organogenesis and a very high instability. They also found that this was correlated with a massive misregulation of DNA methylation. Later on, they produced a line where all 3 genes were genetically knocked down by a tDNA insertion, and this line turned out to be viable. So I'm going to tell you more about this line, which has lost the expression of the 3 H1 variants in Arabidopsis. So I'm showing you now here a nucleus, which is inspected without any fancy microscopy, just a white field microscope, nucleus stained by DAPI. And in the presence of H1, we see the regular aspect with clearly defined chromocenters, and between 6 to 8 chromocenters. And in the absence of H1, what we see is that we have a massive decondensation of the chromatin with only and barely 2 chromocenters. This was the first level of analysis, which already reminded us of what had been described in mouse. In mouse, this is a typical cell nucleus with very strong and bright chromocenters, and in the absence of H1, chromocenters do not totally disappear, but they are massively reduced. And in mouse, it is lethal. So we went a little bit further in the analysis and thought white field is not enough to understand the phenotype of Arabidopsis cells, which are lacking linker histones. And we went for super resolution microscopy, so called STED in that case, so stimulated emitted depletion microscopy. It allows to go down in resolution to about 50 to 100 nanometer, depending on the adjustment. So what you see on the left side is a typical nucleus from wild-type tissue, and on the right side, a typical nucleus from Arabidopsis tissue, which is lacking linker histone. What you can already appreciate visually is that without linker histone, we lose really dramatically the formation of heterochromatin. On top, you have the native DNA staining, 3D reconstruction, and below what you have are 3D reconstruction with where the heterochromatin has been segmented. It also quantifies the lack of heterochromatin chromocenter in the nucleus, and there is a very classical measurement which is done called measurement of the relative heterochromatin fraction, or RHF. And this graph is just to show you that we can quantify this loss of heterochromatin formation, and it's diminished by 2/3 in the mutant. So we obviously had a very strong heterochromatin phenotype, and I have to say here that it was only half of a surprise, because it was already known in other organisms that H1 is indeed necessary for assembling a large heterochromatin structure. And the group of Andrzej Jemanowski had already shown that actually if you overexpress H1 in tobacco, you can actually ectopically form heterochromatin. But we were additionally interested to know whether H1 had also a role in euchromatin. And for this, we also looked a little bit more closely at the images that we produced by STED microscopy. Okay, so what you see on this moving plane here are zooms in euchromatin, and the glowing structure which is appearing are chromatin fibers. So this inlet represents only about less than a micrometer, I think about 800 nanometer, one of each of these inlet. Now, it was still not sufficient for us, or not very easy to quantify the distribution of euchromatin on these images. So we went to the ultra-structural level, and we also decided to quantify the distribution of chromatin density. So what you have here on each inlet, on the left side on the wild type and on the right side of the mutant, what you have are images representing the stained chromatin, and the region which are staining the strongest will represent nucleosome clusters. So we have here inlets which represent 500 nanometer times 500 nanometer region of interest in the nucleus. So we really start now to visualize individual nucleosome clusters. So one can even imagine that each of these individual dots represent a nucleosome. And what you can see from these images is that the chromatin has different density. It is not uniformly packed. But what you can also appreciate is that in the triple mutant, you have more space between these densely staining regions. So the space here will be colored in blue or green. So it gives an impression of fluffiness in the triple mutant chromatin. Now, how to quantify the fluffiness is a little bit complicated, but there are actually mathematical functions that allows to describe the spatial autocorrelation of chromatin density distribution. And to make it short, you can end up with a single value which will measure the dispersion of chromatin density. The higher is this value, and the more dispersed and the less regular will be chromatin packing. So we can nearly associate this value D as a proxy to entropy in chromatin density distribution. And what you can see is that in the triple mutant, we have a strong gain in this dispersion or entropy distribution in chromatin density distribution. And we can restore this effect by complementing the triple mutant line with a single H11 variant. I would like to point out here that this higher entropy or this higher disorder that we would see in nuclei which are lacking H1 is very similar to what has been measured in cancer cells in animals. So the same effect is seen in many cancer cell lines, and we have a similar effect in terms of this altered chromatin density distribution. Now, when we look at the molecular level, H1 depletion also has very interesting or even, I would say, unexpected effects. So in this experiment, we have been mapping the nucleosome density along the different genomic regions, and we specifically looked at the expressed loci. On the graph, what you have is a plot showing the nucleosome density among genes which are either highly expressed, so the dark blue line, or which are highly expressed, which are now in the orange line, or genes which are poorly or not at all expressed, which are the blue line. So you have now different classes from highly expressed to poorly expressed. And what you can immediately see is that in a wild type situation, you have clearly different density in nucleosomes depending on the classes of gene expression. So we obviously have clear structural signature which associate with the different strengths of gene expression. And in a way, this seems logical. The more the gene is expressed, the more you need to have the way free for transcription machinery, and it makes sense to have rather little or a few nucleosomes as possible on the way, or at least to have them in a very dynamic way. Now, when you look at H1, we completely lose these structural signatures. Most of the gene categories, whether they are highly expressed or poorly expressed, and I will show you later that there is not much change in the mutant. So whether the genes are poorly expressed or highly expressed, we don't have a very strong differentiation in terms of nucleosome density landscape. So it was quite unexpected, but it clearly shows that H1 also has a role in structuring euchromatin. So beyond this structural aspect, we also came along a surprising finding. We were actually monitoring the abundance of different histone modification in the mutant. So we actually screened several histone modification on the tail of histone 3 or on histone 4, and we checked different residues. And we stumbled across one particular histone marks, which was drastically reduced in the mutant. And this is a very famous mark, because this is trimethylation of lysine 27 on H3. And this mark is associated with many developmental processes in plants. It is controlled by the polycomb group repressive complex, among them very famous member like Curly Leaf or Media, Swinger, MSI, and many groups are working on this polycomb group repressive complex. So we were surprised to see such a massive downregulation of these marks. You can see it here by immunostaining. We are really losing massively the antibody signal in the mutant. This is something we have repeated many times. We have controlled it against other antibody. We quantified it, and there was really no doubt. It was also confirmed by Western blot in the entire extract, even though here the reduction appeared a little bit less strong on Western blots and it appeared on antibody staining, but it was confirmed. And it was confirmed using a third method, using ChIP-seq, which was normalized using a spike-in technique. This work has been done in the lab of Freddy Barnage at the EBENS in Paris. And what the plot is showing you is below, so at the bottom of the graph, that there are about 4,000 loci which are really losing this mark. So it was really unexpected. We did not thought about this connection, and it was raising even more questions. I will tell you why in a minute. Now we still thought of a model. How comes that H1 would influence at all the distribution of H3K27? And we thought of a model where the chromatin folding, which was imposed by the incorporation of H1, would actually favor the propagation of K27 trimethylation by the polycompressive complex 2. So it might simply be a kind of an opportunistic configuration which allows PRC2 to progress around the target loci. Now, why do we think of a model where H1 is involved in maintenance and not initiation? Well, we have enough evidence now that if we completely abolish the establishment of H3K27 trimethylation, the plants are not able to develop properly. And plants lacking H1 actually do develop rather normally, with the exceptions of some phenotypes that I will show you later. So we rationalize that the establishment of this important developmental mark was normal in the 3H1 mutant. However, probably the propagation and maintenance over time was dysfunctional. So all of this is really nice. Actually, we find really massive phenotype in heterochromatin. Heterochromatin is not at all compacted and folded as normally. We see really strong phenotype in Euchromatin with more entropy and disorder in chromatin density distribution. We find also a blurring in the nucleosome density landscape. We also find massive reduction in H3K27 trimethylation, an important developmental mark. So we would expect really massively disturbed plants, but it's not at all the case. And this was really surprising. So on the right side, what you can see are fairly normal plants, fairly normal looking plants. They have leaves, they have stems, they have flowers, they are actually fertile. They have apparently a regular development. And if you profile the transcriptome of these plants, well, accordingly, you find very little alterations. In fact, we see only 700 genes which are altogether either upregulated, so that's now the pink fraction, or downregulated, and that's a green fraction. When we also look at the transposable elements, because remember the heterochromatin is completely decompacted, we could expect that transposable elements are now de-repressed like crazy, but it's not the case. There's only 1.5% of them which are upregulated. So we were actually looking for additional phenotypes who were a bit persistent, and we looked especially during developmental transition. And accordingly, we found some subtle phenotypes in plants lacking H1. So the triple mutant is actually flowering much earlier than the wild type. We can also notice it by the number of leaves at bolting. And this early flowering phenotype is also restored if we reintroduce H11 variants. Triple mutant also tend to produce a bit more lateral roots than in the wild type. So they have a higher frequency of lateral root initiation. It is another developmental transition into root. Seed dormancy is also affected in the mutant. And we have a kind of a clear effect, especially in very young seeds after harvesting. What we also have is a stomatal lineage, which is not very stable. So we still have a proper density across the leaf, but what we have is a higher frequency of clustering of the stomata. And that's usually a phenotype associated with a lack of stability in the stomatal lineage. We also know that the stomatal lineage is controlled by the PRC2 function. So it's an interesting correlation. What we also found is that the mutant, 3H1 mutant, also develops much more root hair. It is another developmental transition in the root compared to the wild type. This is also an effect that we can rescue with incorporation of an H1 variant. Another intriguing observation was the fact that sometimes the root hair are actually dividing. So they are not stable in their root hair identity, which would be a single root, which is stopping cell division. And eventually, sometimes cell division resumes, and we can observe more cell nuclei in the root hair. So collectively, this told us that the plants are doing fine, globally speaking. But when you look at subtle developmental transition, they don't seem to be very robustly regulated in the absence of H1. So altogether, with what I have shown you about the chromatin phenotype and about the plant phenotypes, especially at the level of transition, I would like to invite you to revisit an oversimplified view that we have about linker histones in plants or in animal in general. But in plants, I think it's quite clear. I think in textbook, we often think of or often read about H1 as a repressor, as a stiffener of chromatin fiber and a repressor of gene expression. But it's not true. So I would actually like to challenge a model which has again been proposed in 2020 based on very good finding, but this model I think is oversimplifying the reality and it's not doing any good in understanding what is the real role of H1. So what is proposed in this model is that H1 is a suppressor of gene expression. Well, I would say that's true, but only for about 500 genes, yes? So what about all the 25,000 genes? And this model also proposes that H1, together with DNA methylation, silences transposon. And again here, yes, that's true, but only for 1.5% of transposable element. So what happened for the other 98.5% transposable element? And a third function of H1, which has been shown, is to actually suppress or prevent intragenic transcription, and especially the production of antisense transcript. And that's a novel finding, which we did not at all address. And it was true or described in the paper for about 60 loci. And if the H1 mutation is combined with a MET1 mutation, it goes up to 1,000 loci. So what I would like to complete in this model is that not only it's true, but it's yes, but, so only for a few genes. And in addition, we know that in the suppression function, H1 acts together with H3K27 maintenance. So this, we have seen an effect for about 4,000 loci. And in addition, and that's a new finding from the lab of Freddie Barnage, we know that H1 actually protects other region of the genome against H3K27 methylation. So you see, H1 is not a simple actor of chromatin organization. It's not only about suppression, and it's not only about facilitating H3K27 methylation, because H1 also protects against H3K27 deposition. And this is especially true for the telomeres, and by doing so, H1 actually contributes to separate the telomere away from the rest of the genome in the nucleus. So I'd like to show that actually H1 cannot be summarized under a single function. And there are definitely different parts of the region which react differently to the presence of H1. We have what we can call H1 dependent loci, where the chromatin, which is structure which is mediated by the presence of H1, will be acting as a dominant control over transcriptional regulation. And then besides, you have a lot of H1 independent loci, which means other factors, let them be epigenetic modifiers or transcription factor, they will prime over H1 mediated structure for transcriptional regulation. And I think this should be really considered in the textbook. So in conclusion, I have shown you that H1 doesn't have a simple repressive and structural role in plants. I mean, it was not only our work, but the work of several other groups, which I've mentioned during the talk. Definitely H1 has a dual role in hetero and euchromatin. It acts at different scales, from the nucleosomal scale up to the heterochromatin domain scale. It provides some structural signature along the expressed loci, contributes to H3K27 maintenance, and H1-mediated chromatin organization can be dispensable and quite largely for transposable element and gene expression. But for some loci, it can remain epistatic over regulation. And the subtle developmental phenotype we observe let us think that it contributes not to the basic developmental process, but rather to stabilize the developmental output. And I think this should echo in our mind with other findings, which is summarized here. So in fact, Arabidopsis, our model plant, is buffering quite well the impact of deficient chromatin controls. And I'm giving here only a few examples. When DNA methylation maintenance machinery is lost, when the plant lamins protein are lost, or some histone methylase protein are lost, plants still develop pretty fine. They might be a bit smaller, they might be a bit larger, but they do flower, they are not really highly sterile and so on. So there is definitely a buffering action. And the same is true for linker histone. So I think we should really revisit our idea of a functional higher order chromatin organization. Obviously, the causal relationship is not that clear. Another possibility is that we look at Arabidopsis in very stable and unchallenging condition. in our growth chamber, and we might measure things in a too well-regulated manner. So what we are maybe missing actually critical situations that would highlight a role for these chromatin modifiers and for H1 in particular. I think we should shift our models from stable to challenging. And as a hint in this direction, we know already that a triple mutant H1 behave really in an antagonistic manner depending on the light regime. So if we have continuous light, the mutant is performing very well in producing calluses or in producing lateral roots. But if we start to grow it in a diurnal rhythm, you could say that you challenge the plant with the day and night changes, then the mutant performs much more poorly. So it might give a hint here of a connection between an important chromatin organizer and the ability for the organism to actually adapt to new physiological situations and fluctuation. So that, I think that's the next roadmap for the few years is to investigate the role of H1 and the factors interacting with H1, not in basic developmental process, but rather in processes that secure smooth transcriptional transitions during physiological adaptation, for instance. And with this, I would like to acknowledge the people who have worked on these studies, and there are many people at the Institute at the University of Zurich who are listed here, especially Kinga Rutovic, who is now authoring several papers on the role of H1. We have a long-term collaboration with the lab of Andrzej Szymanowski in Warsaw and with the lab of Freddy Barnesh in Paris. And this work is not possible without funding, of course, and several organizations have led to the secure funding of the experiment throughout the work. So I hope you enjoyed this webinar, and if you have any question about our work, feel free to email me anytime. Thank you for your attention.